UV visible spectroscopy or electronic absorption spectroscopy. UV visible spectrum results from the interaction of electromagnetic radiation in the UV visible region with molecules, ions or complexes. So you can see that this is the electromagnetic spectrum, and in this electromagnetic spectrum the UV region is what we are studying. So this will give you, in this region you will get the UV visible spectrum. So from the molecular orbital diagram, there are different possible electronic transitions that can occur in each of the molecules with the different relative energies. And the molecule that absorbs energy, that electron can promote from an occupied orbital to an unoccupied orbital of greater potential energy. For most of the molecules, the lowest energy occupied molecular orbitals are the sigma orbitals, which corresponds to sigma bond, and the pi orbital that lies at somewhat higher energy level, that is the pi orbital. This one is a pi. And then comes the unshared pairs that holds the orbitals that hold unshared pairs, that is the non-bond. You can also see the presence of unoccupied or antibonding orbitals like pi star and sigma star also in the case of electronic transitions. So although the UV spectrum extends below 100 nanometer, that is a very high energy, oxygen in the atmosphere is not transparent below 200 nanometer. So special equipments are needed to study vacuum or far UV region. So the routine organic UV spectra are taken between 200 to 700 nanometer. So this limits the transitions that can be observed. So sigma to sigma star, sigma to pi to pi star, n to sigma star, n to pi star. These are the different types of transitions that are observed. But all these are not possible. These are only possible with something called selection rules are following. For an electron to undergo transition, certain quantum mechanical constraints should be applied, and they are called selection rules. So when an electron that cannot change its spin quantum number during a transition, that means they are forbidden transitions. Otherwise, it is a allowed transitions. So forbidden transitions are sometimes observed with the low intensity because of various other factors. So as you can see, these are the different transitions that are observed. Here the n to pi star is a forbidden transition. N to pi star can be a forbidden transition. And a sigma to sigma star electronic transition that involves the electron transfer from a bonding sigma orbital to a sigma antibonding orbital. The transfer of bonding pi electron to an antibonding pi star orbital is the pi to pi star transition. So when one of the electron of a non-bonding pair of electron, that is n, undergo a transition to the pi star, that is n to pi star transitions. And you can also observe sigma to pi star transitions. So these n to pi star transitions that we are seeing here, this always occurs when the molecule encounters should have a heteroatom or multiple bonds. Example, C double bond O, C double bond S, C double bond N, N double bond N, etc. So these electronic transitions that we are seeing is undergoing in a particular type, like sigma to sigma star is having highest energy, then comes sigma to pi star, then pi to pi star, then sigma to n to sigma star, and n to pi star. Now, how to get a spectrum? How can we get a UV visible spectrum? For getting that, the sample that we have to study is irradiated with the electromagnetic radiation over a range of wavelength, and a monochromatic radiation. That is, monochromatic radiation means a radiation of a single wavelength is employed at a time, and this process is called scanning. So the amount of radiation that absorbed at each wavelength is measured and plotted against the wavelength to obtain a spectrum. So a typical UV visible spectrum is the plot of wavelength or frequency versus intensity of absorption. So this is one of the examples of a spectrum. In this case, the first case is a case in which your sample is in gaseous or vapor phase. So the spectrum is very closely packed. In this case, it is in a solution phase. That means the absorbing species are surrounded by solvent molecules, and these solvent molecules, because of the presence of this, they undergo constant collisions with them. And these collisions and the interactions among the absorbing species with the solvent molecules can cause the energies of the quantum states to spread out. So as a result, the photons spread over a range of wavelength, and you obtain a spectrum with a smooth and continuous absorption peak like this. So these are the two different possibilities. This is a gaseous phase. This is in the solvent phase, when solvent is present. So there are three factors which govern the amount of absorbance exhibited by a sample. The thicker the sample, the more absorption takes place. And when the sample is more concentrated, more absorption can be takes place. And some molecules are better than others at absorbing a particular wavelength. So it is an inherent property of the molecule. So these are the three different factors by which absorbance are depending on. Now this is what are the characteristics of a spectrum? This is a typical spectrum, and you have got x-axis wavelength. Sometimes we also use frequency, and y-axis you have absorbance. Sometimes we use intensity of absorption. And this UV spectrum of substances are characterized by three parameters. First one is the position of the maximum of the absorption band, that is what we call lambda max. Second is the intensity of the band. Third is the width of the band. So what is the position? What is position? And the lambda max are referred to. Lambda max is the they refer to the wavelength of the most absorbed radiation, and is a measure of this difference between electronic energy levels that is involved in a transition. And intensity means it is a probability of the transition, that is whether the transition is allowed or not. It is also the measure of a concentration of the absorbing species. And the third one is the width. That depends on the type of instrument that we are using, and also the intrinsic nature of the species that we are being studied. So this is a UV visible spectrum of acetone. You can see there are two bands, one around 187 nanometer and the second one around 270 nanometer. And acetone, from the structure we know that acetone has got pi and non-bonded electrons in it. So there are two possible transitions, N to pi star and pi to pi star. As you can see, the red one is N to pi star transition and the blue one is pi to pi star transition. N to pi star transition is having lower energy, that means higher wavelength, and pi to pi star transition has got higher energy, that means lower wavelength. Now coming on to the effect of conjugation on wavelength. Is there any effect of conjugation in a wavelength of a particular compound? This is acetone. Acetone, you have lambda max around 187 nanometer and 270 nanometer. And this is another compound, methyl vinyl ketone, that is having lambda max around 219 and 324 nanometer. So from acetone to methyl vinyl ketone, there is a shift in the pi system. There is a more extensive pi system in the case of methyl vinyl ketone. As a result, the N to pi star transition, the pi to pi star transitions observing in methyl vinyl ketone has got longer wavelength as compared to acetone. So now what are the effects of conjugation? Conjugation, when a molecule is having more conjugation, that means the energy gap between HOMO and LUMO decreases. That means it is having less energy required for the electronic transition. That also indicates that the transition occurs at longer wavelength. Lesser energy means longer wavelength. If a compound has enough double bond, what will happen? It will absorb visible light and the compound will be colored. For example, beta carotene is an example, which is orange in color. That can be observed in carrots and tomatoes. This is the structure of beta carotene. It has got lambda max around 455 nanometer. So colors can be obtained in the case of if the visible, the light is that we are giving is able to absorb visible region. So that's about UV visible spectroscopy. Thank you.